Diffusion by Levan. Diffusion, definition and purpose. Metabolic pathways require the participation of molecules that must move across membranes and through cells. Diffusion is the spontaneous spreading of molecules or ions, and it is an essential way substances move into, through, and out of cells. Atoms and molecules are always jiggling, and this internal movement causes it to randomly bounce off objects, including other atoms or molecules. Rebounds from these collisions propel solutes through a liquid or gas, resulting in gradual and complete mixing. How fast this process occurs depends on 5 factors. The 5 factors of diffusion are 1. Molecular size, 2. Temperature, 3. Concentration, 4. Charge, 5. Pressure. One, molecular size. It takes more energy to move a large object than it does to move a small one, so small molecules diffuse more quickly than large ones. 2. Temperature. Atoms and molecules jiggle faster at higher temperature, so they collide more often. Thus, diffusion occurs more quickly at higher temperatures. 3. Concentration. A difference in solute concentration between adjacent regions of solution is called a concentration gradient. Solutions tend to diffuse down their concentration gradient from a region of higher to lower concentration. 4. Charge. Each ion or charged molecule in a fluid contributes to the fluid's overall electric charge. A difference in charge between 2 regions of fluids can affect the rate and direction of diffusion between them. For example, positively charged substances, such as sodium ions, will tend to diffuse toward a region with an overall negative charge. 5. Pressure. The rate of diffusion may be affected by a difference in pressure between 2 adjoining regions. Pressure squeezes objects, including atoms and molecules, closer together. Atoms and molecules that are more crowded collide and rebound more frequently, so diffusion occurs at higher pressures. Semipermeable membranes. Lipid bilayers are selectively permeable membranes. Water, gases, and hydrophobic molecules can cross them, but ions and most polar molecules cannot. When 2 fluids with different solute concentrations are separated by a selectively permeable membrane, water will diffuse across the membrane. The direction of the movement depends on the relative solute concentration of the 2 fluids, which are described in terms of tonicity. Isotonic fluids have the same overall concentration. If the overall solute concentrations of the 2 fluids differ, the fluid with a lower concentration of solutes is hypotonic. The one with the higher solute concentration is hypertonic. The movement of water across membranes is known as osmosis. Semipermeable membranes continued. If a cell's cytoplasm becomes hypertonic with respect to the fluid outside of its plasma membrane, water will diffuse into the cell. If the cytoplasm becomes hypotonic, water will diffuse out. In either case, the solute concentration of the cytoplasm may change. If it changes enough, the cell's enzymes will stop working, with lethal results. Many cells have built-in mechanisms that compensate for differences in solute concentration between cytoplasm and extracellular fluid. In cells with no such mechanism, the volume and solute concentration of cytoplasm changes when water diffuses into or out of the cell. Passive transport. Osmosis is an example of passive transport, a membrane-crossing mechanism that requires no energy input. The diffusion of solutes through transport proteins is another example of passive transport. The movement of the solute and direction of movement is driven entirely by the solute's concentration gradient. Some transport proteins form pores, permanently open channels through a membrane. Other channels are gated, which means they open and close in response to a stimulus such as a shift in electric charge or binding to a signaling molecule. Facilitated diffusion. With a passive transport mechanism called facilitated diffusion, a solute binds to a transport protein, which then changes shape so the solute is released to the other side of the membrane. A glucose transporter is an example of a transport protein that works in facilitated diffusion. This protein changes shape when it binds to a molecule of glucose. The shape change moves the glucose to the opposite side of the membrane, where it detaches from the transport protein. Then, the glucose transporter reverts back to its original shape. Active transport. Inactive transport. A transport protein uses energy to pump a solute against its gradient across the cell membrane. Typically, an energy input changes the shape of an active transport protein. The shape change causes the protein to release a bound solute to the other side of the membrane. An example of this is when a calcium pump moves calcium ions across cell membranes by active transport. Calcium ions act as potent messengers inside cells, and they also affect the activity of many enzymes. Thus, their concentration in cytoplasm is tightly regulated. Calcium pumps in the plasma membrane of all eukaryotic cells can keep the concentration of calcium cells in cytoplasm thousands of times larger than it is in extracellular fluid. Another example of active transport involves sodium-potassium pumps. Nearly all cells in your body have these transport proteins. Sodium ions in cytoplasm diffuse into the pump's open channel and bind to its interior. A phosphate group transfer from ATP causes the pump to change shape so its channel opens to extracellular fluid where it releases the sodium ions. Then, potassium ions from extracellular fluid diffuse into the channel and bind to its interior. The transporter releases the phosphate group and reverts to its original shape. The channel opens to the cytoplasm where it releases the potassium ions.